Hello, everyone, and welcome to today's webinar, Widely Applicable Charge Variant Analysis of Therapeutic Proteins, presented by Peter Holper, Application Scientist, SCIEX. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more about our sponsor, please visit sciex.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Peter Holper, Application Scientist at SCIEX. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Peter, you may now begin your presentation.
Thank you very much for the introduction. Prior to joining SCIEX, I had many years as an analytical development chemist who, like you probably are now, was always searching for ways to improve my workflow, ways to improve my methods. And at the end of the day, I was really looking for ways to shorten my workday. I wasn't in the lab still running samples at 6 o'clock while my family was eating dinner. Work-life balance was always very important to me, just as I'm sure it is to you. So I'm excited to be here to share a potentially new way to analyze charge variants that also has applicability to a wide range of molecules. Now, as an analytical chemist, when I would hear someone talk about a charge variant method that could work for a lot of molecules, I would probably envision a 90-minute pH gradient ion exchange method. But I'm going to assure you, we're going to be discussing something far more appealing. I'm looking forward to helping you improve your future and just get out of the same way you've done things in the past. So we'll start today with a brief introduction to overall analytical control strategy of therapeutic proteins. And we'll touch on how those methods that govern our analytical control strategy can themselves be developed through a quality by design approach. Because without quality in our method, we don't have a method, because everyone would always question the result. And after that, we'll do a deeper dive into charge heterogeneity method development, focusing on finding that widely applicable charge variant analysis method. We'll follow that with just a brief summary. So when I was in your shoes, tasked with developing an analytical control strategy for a therapeutic, my first consideration was always, what are the regulatory agency expectations? And this really set the stage for developing the methods that were going to separate out the molecule's quality attributes and its CQAs. These areas included methods to quantitate fragments, methods to quantitate aggregates, charge variants, and potency. But when I started out, I was very much a part of a groundswell of new molecules. We'd go through churning through maybe 100 molecules through our whole department to multiple hundreds with less resources and bigger time constraints. And so when we think about that, we're going through so many compounds, developing assays for all these compounds when maybe a fraction of that will make it to IND, and a fraction of that will make it to market. We still have to put in all that time, all that effort at the beginning to make sure we have quality methods. We would always try to leverage what we could. Template methods, our go-to, really universal methods. Charge variants never really fit that bill. Charge variant method development was always the most time-consuming and unique. But why? Why was that the case? Well, that's the case because each molecule that we work with has a unique amino acid sequence. And with that unique sequence, the potential modifications to that sequence are going to be unique. Well, one molecule you work with may be very prone to fragmentation. Another coworker is going to be easily oxidized on a histidine or a tryptophan. And when we think about all those different charge modifications, it quickly becomes evident that developing a common universal method to be able to really robustly quantitate all these different charge variants would be a near impossible task. And what does this lead to? This leads us to spending an enormous amount of time in the lab developing many individualized methods for each specific molecule. And with more molecules in our pipeline compared to last year, the year before that, 10 years ago, and with less time to do it and less starting material, that's just more and more of a daunting task. I remember just a few years ago, we could submit our IND without a specification for charge variants. But that's in the past. Now that specification is an expectation. And so we're tasked with generating specs on our methods that has maybe only been used on a couple lots of material, maybe a development lot and a tox lot. So we're developing a robust method earlier in the timeline with less material, all while doing our normal daily work. And we want to make it home in time for dinner. Or if we're lucky, maybe even to make dinner. We know our methods would not be considered a control method if it wasn't robust. So how do we start to develop that robust method? Well, we can start with a quality by design approach. And as you may know, quality by design exists to build robustness into our method development. So we aren't caught needing to redevelop the method further down the line just due to a problem we didn't initially encounter when we were developing the method. And I really enjoy this quote because it rings so true in a time where we are pushed to bring molecules to market faster and are given less time and less resources to make it happen. And that quote is this: "There's never enough time to do it right the first time, but there is always enough time to do it over." And that is what quality by design seeks to eliminate. Doing something over because we've done it right the first time. So the goal of our analytical method development here is twofold. We want to ensure our patient safety and our molecule efficacy by understanding where in the process our PTM CQAs are coming from. And then we want to help support our manufacturing process that will minimize them from occurring. Our second goal, and the real big challenge we hope to overcome, is to define a control method that can take these large, complex, diverse populations of molecules, successfully separate and resolve the modifications so that we can answer really the first goal of where is that PTM coming from. And our quality by design approach can help achieve these goals. Now, this approach is driven by the analytical target profile, which is going to define the objectives of our assay, of our test, and the quality requirements of the data that we're producing. So for a charge variant method, we would expect or require there to be no interfering peaks from matrices or buffers in the region of interest where our sample will migrate or elute. We would also expect it to be robust and still stability indicating, so that any changes we see from lot to lot or on stability are truly indicative of the molecule and not the method. We don't want our other groups coming to us and not believing the data we're providing them. But there are also other attributes we have to consider. One of them just being business drivers. What's the cost of running the method? If we're churning through a $2,000 column every couple runs, that's not a good method. What's the time required to run the method? Because I, like you, would never want to run a 90-minute method where I have to start it, wait for an hour and a half just before my standard is injected. How about the supply of the reagents used? Do you have multiple vendors that can supply it? And what's the applicability to different labs, both near your own potentially in-house QC or quality labs at different countries where the molecule might end up when it's commercialized? If they don't have your equipment to run it, then you can't sell there. Well, another consideration are method attributes. What does a good method look like, especially for charge variants? Are we looking for the profile with the most peaks? Are we looking for the profile with the best peak resolution? Or should we just start with a method we expect to have the lowest development time so that we can make it home and leave at 4 o'clock every day? We don't know going into it, and that's what makes charge variants so difficult. So to meet the analytical challenge that our molecules pose to us, we have different options, different techniques. 2 of those options are ion exchange, chromatography, and SCIEX. Now we'll take a quick look at these and more as we search for a widely applicable method. And we'll start with a very commonly used charge variant technique, ion exchange chromatography. Now, many of us are familiar with ion exchange chromatography because we're familiar with using an HPLC. That might be the first thing that we learned going into an industry lab. And if we think now of just cation exchange chromatography, we have a positively charged surface residue on our molecule that are binding to a negatively charged stationary phase. Now, how strongly the molecule is going to be bound to that stationary phase is dependent on the strength of each bond and the number of interactions between the molecule and the stationary phase. Well, the molecule can be eluted off with either a salt gradient or a pH gradient. We have to think now. Just because it's very commonly used doesn't make it the best choice for each molecule we're analyzing. And in fact, there are a lot of parameters beyond just these that we should investigate to ensure it's properly optimized. And since we don't know the stationary phase and how it's going to interact with our molecule, there are a lot of choices out there, a lot of different vendors. So when we think of the parameters that go into building an ion exchange method, we should evaluate the buffer salt, the ionic strength of that buffer salt, the buffer pH, whether we're using a salt gradient or a pH gradient. We should evaluate the flow rate. We certainly need to evaluate the column temperature, and as I mentioned, the column manufacturer, because each resin, each stationary phase is going to be different. And as I list all those parameters that we should evaluate, remember, there is never enough time to do it right the first time, but there's always enough time to do it over. And I found it rare, if ever, to have enough time in my day to really thoroughly investigate all these parameters for multiple vendors. I'm sure you can agree. So if we look at what profiles might result from all that work that we did, that we put in there, weeks, months potentially of work, they're no doubt going to vary depending on the conditions we used. So first, we should be screening multiple stationary phases to find the best profile, what we consider the best profile anyway. From there, we need to determine whether a salt or pH gradient offers the best resolution for this one molecule. And once we settle on those parameters, we should really run a little DOE to ensure we have a robust method, ensure we're in a robust space, so that when the next person picks it up, they get a nice, good result, just like the one we got. Now, all told, right, this could take us weeks or more of work for each new molecule coming through the pipeline. There's no guarantee that this is going to work more than the current project you're on. In fact, it probably won't. The next molecule probably won't work at all. That's a lot of work invested for one molecule very early in the process. And above all that, this might not even be the best method you can achieve. If we look at this profile here, the result looks pretty okay after weeks or more of work. There has to be a faster way to get the answers we need. Nobody wants to be in the lab at 7, 8 at night. So the second analytical technique option we'll cover on our search for really a widely applicable charge variant method is SCIEF. In SCIEF, the analyte we're analyzing is going to be separated by differences in its isoelectric point. So we'll have a master mix containing a mixture of amphiolites, additives, PI markers, all mixed in there with our sample before it's injected into our capillary. And once our electric field's applied, the proteins, the amphiolites are going to focus in that capillary based on its PI. Now, compared to ion exchange, this seems like a fairly straightforward technique. But just like we had with ion exchange, SCIEF also has a number of development parameters we really need to optimize to ensure we have a really good method for that specific molecule. So we're going to look at different amphiolite manufacturers, different mixtures of amphiolites, different ratios of those amphiolites, various additives in varying concentrations, and finally, the amount of focusing time of our method to really ensure that molecule has been fully focused prior to being detected or prior to taking that image. So if we look at a profile that might result from that optimization work, it again is going to vary depending on the additives used, right? Depending on the amphiolites, amount of ureas or other additives. So if we use a denaturant and really a broad range amphiolite in this profile, we can see nice resolution of the basic variants, the C-terminal lysine basic variants, and some resolution of the acidic variants. Now from here, we'll probably say, let's screen some other amphiolites, let's screen some other additives, so we can do a little bit better. And then we're going to end up with more profiles. So from that work, if we focus down on using really a narrow range ampholyte and some additives in there, we can certainly achieve increased resolution of both the acidic and basic variants. Once we've settled now on those optimized parameters, we'll run a little DOE, make sure we're in a good space, very robust space, so that when we send it to our CRO, our results are comparable. And when I was learning IEF, and as I'm sure many of you over the past years have, it seems like a fairly universal technique at first. But I quickly realized that a lot of molecules in my pipeline, a lot of molecules that I worked with, just didn't quite work with these conditions. It didn't look good. It wasn't very robust. So I still had to do a lot of work. I still had to make a lot of individualized methods for each molecule. I was still in the lab. I mean, I love developing methods in the lab, but I also love eating dinner on time. So really, beyond ion exchange and CIEF, what's left? What else is out there that we can try? We need a method that will really work for a lot of molecules, just right out of the box. And we really need a technique that doesn't have just a multitude of parameters to optimize, because we just don't have the time. We don't have the resources. We have management constantly getting on us, project managers, timelines, hit that timeline. But we know we need a robust method, so there's a give and take. We need a technique that is just easier. And since we don't have the time that we need to do all that necessary method development, we start to think, well, what if the method development portion was already done for me? That would make my job a lot easier. I would love that. And so today, that's what we're introducing. Our new CZE kit for protein analysis, for charge variant analysis. And this is the CZE Rapid Charge Variant Analysis Kit. As you would expect from a CE method, it's very high resolution. And this type of separation, really a nice benefit in my mind, is that it's done in native analysis. So we're not denaturing, as we generally are in IEF. So we're analyzing the molecule as it is. And the sample prep is non-existent. It's just dilution in water. That's as easy as size exclusion, chromatography. Right? The method development is already done for you. The separation buffer itself coats the bare fused silica capillary, which really just makes the method a breeze. The capillary, the bare fused silica capillary, is maintenance free. The sample buffer that we're using is just water. The method, the technique, is extremely easy to use, extremely fast. The separation can occur in 10 minutes, even with rinsing steps. That's even faster than ICIEF. It really is that easy. As a former person that used to run this method in the lab, it really is that easy. So, how does it work? Well, CZE is the simplest type of capillary electrophoresis separation, and it separates analytes based on differences in their mobility, which are roughly a function of the analyte's charge to hydrodynamic radius ratio. And it's a very simple technique because all that's needed for the method is a well-chosen buffer. And while it is that simple, it can also be very powerful. So when the size distribution, the hydrodynamic radius of your sample, is fairly negligible, as is the case with most antibody samples and other proteins where we have good control over our manufacturing process. The separation is primarily based on differences in the molecule's charge. Now, even with a short analysis time, we can still achieve very significant resolution of charge variance. Using the NIST map in this example, we see an excellent separation of both the acidic and basic variants, along with a very reproducible profile shown on the right, where we're just overlaying 12 individual analyses. And what's even better is that you can be really confident in the method's reliability and its robustness, as it's already been validated across multiple sites, with many of the participants being the companies you work for that participated in this evaluation just a few years ago. Now, CZE can work with a wide range of molecules, as I mentioned. So we're going to show you 3 commercially available monoclonal antibodies, all from different companies. All of them were separated using the pre-assembled cartridge and the CZE Rapid Charge Variant Analysis Kit on the P800 Plus. And the only preparation that was involved was a dilution of the molecules to 1 mg per mil in CE-grade water. The first molecule is trastuzumab, under the commercial name Herceptin. Right? So in 10 replicate analyses of this very well-resolved profile, we achieve RSDs of less than 1.5% for total acidic variants, total main peak, and total basic variants. Now we'll look at rituximab, better known as Rituxan. Again, 10 replicate analyses, another very well-resolved profile, with RSDs of less than 2% for total acidic variants, main peak, and total basic variants. And finally, we'll look at infliximab, commercial name being Remicade. Here again, 10 replicate analyses, an incredibly well-resolved profile is achieved, with RSDs of less than 2% for total acidic, total main peak, total basic variants. When we're tasked with developing an analytical charge variant method, we're presented with challenges that are unique to each molecule. And those can include solubility issues, challenges around peak resolution, unique PTMs. Well, CZE and the SCIEX Fast Charge Heterogeneity Analysis Kit has many advantages over ion exchange and CIF to meet those challenges, including a low development time, very high peak resolving power, and very fast analysis time. And because robust method development is always our goal, and because we never find ourselves with an overwhelming amount of time to really dedicate to it, we now have another tool, the SCIEX Fast Charge Heterogeneity Analysis Kit. We have our robustness built in. We have our QBD already in there. And it's all there for you on the P800 Plus platform, which features sample temperature control, capillary temperature control, which gives you good sample stability, your migration time reproducibility. We also provide the Easy CE pre-assembled cartridge for ease of use and traceability. Got the lot number printed right there on the front. So all these features are available on an instrument and a technique that has been validated across the industry. really proves its unmatched reliability and robustness for your needs, for your day-to-day needs. And with that, I would like to thank everyone for taking the time to listen to me today, and I would like to open it up for any questions you may have.
Thank you, Peter, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, Peter, let's get started. We have lots of great questions already coming in. Let's start with this first question. Can CZE work on molecules other than mAbs?
Yeah, certainly. CZE is kind of nice and unique in that because we're not looking at or really relying on any interaction between the surface of the molecule and the stationary phase, we aren't relying on it being a certain size. So I've used CZE personally on mAbs. I've used it on certain fusion proteins, even viral viruses. Some of our customers have used it on bispecific antibodies, probably other fusion proteins than what I had worked on. And what's really just important is to make sure that it's charged in solution. So generally the molecule's PI is going to be what drives whether or not we have a charge and can migrate that through the capillary. But the construct of the molecule itself doesn't hold much significance, so it can work with a wide range of molecules.
Thank you, Peter. Our next question. What type of molecules are more suitable for a CZE approach over ion exchange or SCIEX?
Okay. So I think in talking with a lot of industry colleagues and even in my own previous research, what we've kind of agreed upon is molecules that are more hydrophobic have a hard tendency to be analyzed by ion exchange. So they have limited interaction with the stationary phase for some reason. You're not getting very good resolution. That can happen a lot with antibody-drug conjugates. So I think that at a high level would be more suitable for a CZE approach. For CIEF, you really need to have a molecule that is soluble in solution. So there are certainly molecules that either don't want to be denatured or you just can't find the right additives for them to stay in solution in CIEF. Those would be an excellent candidate for CZE. But really, in my experience, any molecule to just throw on this rapid charge heterogeneity kit is great because it's so fast. So it's very easy to screen, much faster than ion exchange or CIEF, just to see if you get a good profile.
Now, Peter, can high levels of matrix salt affect a CZE separation?
Okay, so I think myself and probably a number of people listening have experienced an in-process sample that completely ruined your CIEF capillary due to the coating. So that's definitely something that we are wary of. For CZE, at least in this case, we aren't using a coated capillary, and since we aren't filling the capillary itself with a mixture of our sample and a master mix in the case of CIF, we aren't injecting an enormous amount of salt or an enormous amount of that formulation matrix. So in general, no, there really aren't, in my experience, there hasn't been much of any impact from high levels of salt in the sample matrix.
Peter, is there an advantage to using a UV detector specified in the CZE method over the PDA detector?
That's interesting. For this particular CZE method, no, you should be able to use either interchangeably. We did do the validation, I believe, a few years ago using a UV detector, and it can be what is available in your lab. But because we're using a bare fused silica capillary, use of the PDA detector It's not going to degrade anything because there is no coating, so I wouldn't expect to have really any difference in peak profile or performance of the assay itself.
Thank you, Peter. Now, we have lots of great questions coming in here. Let's go with this next one. Are there ways to increase resolution?
Certainly. So what I think is nice about how this kit is offered is because it doesn't have a capillary cartridge included in it, you can use whatever length of capillary and capillary cartridge you want. So you're not limited to a 30 centimeter capillary. You can go all the way up to, say, 60 centimeter if you want. And very analogous to an SEC separation. If you put two SEC columns in series, you're going to get a slightly increased resolution. Well, the same way with CZE, if we give the molecule more time to migrate through, you're going to get more resolution, more separation between the different variants of that molecule.
Peter, would CZE be useful for molecules in the discovery stage?
Yep. So again, in my previous work, we worked closely with discovery. We were looking at, or they were, I should say, looking at charge variants as one of the early, I guess you'd say, a developability assessment. And they did a little work with ion exchange, which proved problematic because it took so long. They also tried IEF, which was a little faster, but they would always run into molecules that really didn't work. And I think CZE is a great approach because, again, of just how simple it is. You just throw it on there. There really isn't anything to make up. The sample prep is a dilution, but in discovery, your titers are probably low enough you might not even need to do that. And you can really get some nice knowledge about your molecule or degradation pathways just by seeing different pH effects if you're keeping it on a short-term stability study or a short-term stress study for discovery.
Thank you, Peter. How many injections will a cartridge last?
Okay, so yeah, this, again, because we're using a bare-fused silica capillary now, a lot of us might have had prior experience with a coated capillary, a neutral capillary, or FC coated capillary, something of that effect that generally we'll say lasts for about 100 runs or could certainly last longer, but that's generally what they are marketed as, at least 100 runs. With a bare-fused silica capillary, I think you could expect to be in the multiple hundreds of runs because there is no degradation of that coating. Now, what will happen is over time with the acid rinses will start to break down and cause the capillary itself to become brittle. But I have heard upwards of 500, 600 runs on these capillaries. It's possible to go beyond that. Certainly we will guarantee it, for probably 100 runs, just like we do our neutrals. But you can, and I would, expect a far longer longevity out of this type of capillary and separation.
Thank you, Peter. And let's go with this question. Does CZE allow ballpark estimation for PL?
So no, it doesn't. So this would be similar to, I guess, ion exchange, where ion exchange can't calculate the PI of the molecule. CZE can't either. You could run the PI markers, but since we aren't forming a pH gradient in the capillary itself, there's no way to assume that you're going to have really any linearity of the markers that you could even spike in there. But from my experience, right, we only need to calculate experimental PI once for IND. So when we have used CZE approaches or ion exchange approaches, we just run it on IEF once. And what's nice is on the P800 Plus system, you can run them both right there. So you run it once for your IND, then you move back to your control method, which could be a CZE approach.
Thank you, Peter. No, Peter, before we go, do you have any final comments for our audience?
Well, yeah, I just want to thank everyone for listening, and I really do hope that you try out the kit. I've found it very useful in my previous positions, and I know a lot of our customers that have gotten to use it are happy with it and have found a lot of usefulness on various molecules that really at the end of the day, we're just trying to save time, work our molecules through the pipeline faster, get home to our kids, our family, and not be stuck in the lab so long sometimes.
Thank you, Peter. And thank you for your time today and your important research. I'd also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. And then one more time before we go, I'd like to thank our audience for joining us today and for your interesting questions. Those questions we were unable to answer today and those that are submitted during the on-demand period will be addressed by Peter via the email address you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you shortly when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. Have a great day.